Welcome to Population Genetics. In this video I'm going to focus firstly on how we use the Hardy-Weinberg equation, and then I've got lots of examples of how it's used within the exam. So first of all, there are a few key definitions that you need to know. The first one will be gene pool. So a gene pool is all of the alleles of all of the genes of all of the individuals in a population at a given time. So it's the idea that they're all mixed together. They're not associated with one person. It's not the idea that someone has a dominant or someone has a recessive. It's the idea that how many dominant alleles do we have and how many recessive alleles we have. Another key definition that you need to know is allele frequency. So this relates to the gene pool, and it's essentially the number of dominant alleles or the number of recessive alleles. So as a definition, the number of times the allele occurs within the gene pool is known as the allele frequency, and we're referring to either the dominant or the recessive. So the Hardy-Weinberg equation is divided into two, as you can see on the right hand side. The initial equation looks at the dominant allele frequency, P, plus Q gives us the frequency of 1. Now Q being the recessive allele. Now all this means is that it's just looking at the dominant allele or just looking at the recessive allele. So the red dot represents the dominant allele and the pink dot represents the recessive allele, and you can see there's only one of them there. It's not this idea that there has to be two present. When we're using the top equation, there can be a single allele present. So the bottom equation is very different. This is looking at the frequency of people or organisms or snails or birds or whatever it is. But if we use the bottom equation, we're suggesting that they have two alleles present. So P squared is the frequency of homozygous dominant. So this is the frequency of people or organisms that have two dominant alleles. 2pq is referring to the idea that they have one dominant and one recessive allele, and then q squared is the frequency of homozygous recessive organisms or individuals. Again, they've got two recessive alleles. The way I remember it is if you face a question which is talking about a cat, a dog, a snail, a person, they have to have two alleles inside of them. And if they've got two alleles, then you're using the equation that has two in it, either p squared, 2pq, or q squared. If it's just talking about the dominant allele frequency or the recessive allele frequency, then it's going to be the top equation that you're using. Frequency is always represented by a 0 point. That tells you that it's a frequency. So in a second I'll show you an example, but before we move on, we have to be aware of some assumptions we assume when we're using the Hardy-Weinberg equation. So there should be no immigration, no emigration. There should be no mutation, no selection. There should be a large population, and mating should be random. Now, this can come up as a recall question, so you simply need to learn them. But if all these assumptions are maintained, it means one really key thing. It means the allele frequency from one generation to the next will always be the same. In reality, this doesn't happen, but if these assumptions are maintained, then the allele frequency will be the same. So we're going to move on to looking at an example of how we use the Hardy-Weinberg equation, and we're going to follow these rules on the left hand side. So we're working from step 1 all the way through to step 5.